this? Okay. All right. Are there any questions before we begin? Okay. So today we're covering the skin system, sometimes known as the integumentary system. So make sure you're keeping up with this stuff in your lab manual. It is pretty histology focused, so that's why we're using the virtual microscopes. And then next time we'll have plenty of time for you to review this in class and work with the microscopes yourselves, so we will not be too far behind. Okay. All right, so you should be familiar with this image from your lab manual. What we're talking about with the integumentary system is all skin and associated structures. So we'll talk a bit about hair. We are not going to cover nails because it's not a huge part of your lab manual. It won't be tested in lab, but we will cover glands, and those tend to be tricky for people, histology-wise. Overall, this guy is our largest organ. It does a lot. So we talked about how squamous epithelium, when it has multiple layers, will help prevent fluid loss from underlying layers. We talked about how those multiple stacked layers also provide mechanical protection to our vascularized underlying layers. It also helps to regulate temperature. So anytime we have many, many layers of epithelium, it has a lot of these protective thermoregulation functions. And we'll also talk a little bit about some of the deeper sensory structures that are involved in sensing temperature, pain, as well as mechanical sensation. So we have three basic layers. The first one, the most superficial layer, is the epidermis. So it's going to sit on top of the other layers, epi meaning above. The dermis is deeper, and it's also going to be a pretty thick layer. That's where we're going to have more of our connective tissue with those collagen fibers we talked about last time. And then we actually have this underlying layer called the hypodermis. So hypo means below the skin, hypodermis. This is not technically part of our skin system. It's rather kind of anchoring the skin to the underlying tissue, which is usually muscle or another layer of fascia. One thing to point out is that the hypodermis has a lot of different names. It's mainly fatty tissue, but sometimes you'll see it referred to as Camper's fascia or superficial fascia. That could be a good lecture question. There's a lot of cells that we're not going to talk about in depth right now. Things like specialized blood cells that are involved in an immune response. But the thing I want you guys to know for this lab is that our epidermis is made of something called keratinized stratified squamous epithelium. So these last three words we already should recognize. We have multiple layers of flattened cells forming our epithelium. Keratinized refers to the hardness of these cells. So keratin is this really tough protein that is produced by cells within the epidermis. And most of the cells we see will be keratinized, which means they're hardened. And they also play a role in our immune defense. So that's one of the reasons why you can get an inflammatory response in your skin to pathogens. It can get red, vasodilated. Same thing with stuff like sunburn. You know, we get an immune response in the skin because we're damaging the cells there. There are other cells such as mast cells, macrophages, those are involved in specific immune responses. We'll talk a little more about these types of white blood cells when we get to blood histology, which is way in the future. Other things you might see are Langerhans cells. So those are specific antigen presenting cells. They're specific to the antigen that they're defending against. So just a little immune stuff, but again, bookmark that for the future. One of the clinical applications I want to talk about is sunburn. So this is technically a radiation burn, which sounds really extreme, but there's varying degrees of sunburn, as I'm sure you're all aware. But what happens is we get this immune response within the epidermis. So you'll get swelling, you can get a lot of redness, that's most common. Sometimes it can even blister. And what's happening is these keratinocytes are taking damage. And if you receive enough damage over a long enough period of time, it can have these permanent mutations. Same deal with these immune cells. So we're actually experiencing an immune response within the skin. The first to act are the mast cells, creating histamines. But chronic exposure can also affect those immune cells, and that's how we get things like this increased risk of melanoma. So just a little PSA, wear sunscreen if you're going to be out in the sun for a while. And let's get into the epidermis. So you want to be comfortable with this image. This is straight from our modules. I like to use this one a lot on the lab exam because it shows the epidermis, the dermis, and the basement membrane really well. So what we have up top, this is our apical surface. So this is facing the external environment. Sun rays would be shining down here onto our epidermis, which is this entire layer or series of layers. A lot of times students are confused about where the basement membrane is. So I want to point out that the epidermis extends all the way down to this wavy pink pattern here. It does not stop at the dark purple. We'll talk about what that layer is, but when you find your basement membrane, look for this wavy pattern on your slides. That is going to be the bottom of the epidermis. Everything below that point, you can see it has this different texture. It's kind of swirly. This is going to be dense connective tissue. And we'll actually have two types we talk about. One thing to note is that thick skin has 5 layers, whereas thin skin only has 4. And most of what we're dealing with is thin skin. Anywhere that you have hair on your body, you will have thin skin. And that will be very important for identifying features on your slides like hair. So here we have a side-by-side comparison of thin and thick skin. There is a nice saying you can use to remember the layers and the order of layers for the epidermis, and that is, come, let's get sunburnt. So the first layer is the stratum corneum. I think corny to me sounds like a hard kind of kernel texture. So I think of stratum corneum as that outside hardest texture that's the first to deal with the environment. So that's going to be this layer up top, most superficial. This guy here, stratum lucidum, is in parentheses because it's only found in thick skin. If we break this apart, lucidum, lucid means light, so this layer appears light on our slides. If we come over here to thick skin, we have that really wide stratum corneum, and directly underneath we have this lighter strip of stratum lucidum that is only present in thick skin. The other 3 layers are standard. So just below the, or deeper to the stratum lucidum, we have this really thin, dark strip called the stratum granulosum. It's named this based on the fact that these cells contain these really dark staining granules, and that's why it appears dark purple on our slides. Just underneath that, we have stratum spinosum, and then our bottom layer is stratum basale. Basal because it's sitting against the basement membrane. So you can use those cues to help you remember the order of these layers. So up top, again, we have the stratum corneum. That's the most superficial. Most of the time, we're dealing with dead cells. This is part of the reason that we can strip off a few layers of the stratum corneum and be totally fine. It's because these cells are already dead. They are keratinized, so they're toughened. They're like, again, I think of like kernels for corn, stratum corneum. And then we have that stratum lucidum, so that tells us that this is a segment of thick skin. Stratum granulosum is that guy containing the dark granules. These last 3 layers, last 2 layers, they start to get a little more active. So the stratum spinosum is actually where we start the process of keratinization, of hardening these cells. And this is that layer, it's just underneath the dark purple stratum granulosum here, and it's getting towards that basement membrane. So most of what we see in this region here is going to be that spinosum. And then the deepest layer is actually the most active. So the stratum basale is that layer really hugging the basement membrane here. This is where cells are being produced continuously. So we actually have stem cells in this region that are just constantly regenerating more epidermal cells. And what happens is as these cells are produced, they start pushing towards this superficial or apical surface. and that is how the epidermis can be regenerated so quickly. So to look at that on a microscope slide, I want to switch over real quick. We'll take a look at the human protein atlas. And let's take a look at some skin. So I like this website a lot because it shows you basically an answer key on the side here. So you can click on any of these elements and see them in further detail. When you're practicing on your own, I would first try to locate things just by clicking around on the microscope, and then you can test yourself by clicking on any of these elements here. So right now, we are just at the level of the epidermis, facing that external environment. If you zoom in, first thing I want you to identify is your basement membrane. So in this case, it's really clear we've got this wavy pattern here separating our cellular epidermis from the underlying dermis. And then from there, you get a little closer, you can see things like this stratum corneum. In some cases, the corneum will start to separate from the underlying tissue. That's just part of the fact that these are dead cells, so they're not going to be adhered to the underlying layers. So you can see that. And then I would say for testing purposes, I would focus more on things like stratum corneum, or if we're dealing with thick skin, try to identify your lucidum, as well as the layers close to the basement membrane. What can happen on your slides is sometimes these layers are not nicely delineated for us. So it can get a little messy once we're at the layer of spinosum versus basala. So again, I would study the things that are distinct and easy to point out under a microscope. Things like stratum corneum, granulosum, and basala. All right. So that was all epidermis. We're going to talk about the underlying, mostly connective tissue layer now. So the dermis has a different function. It's not protective so much as it is providing structural integrity. So what that means is it's going to help adhere the epidermis to underlying layers like our Camper's fascia or our hypodermis. There's a lot going on, as you can see here. We have these blood vessels in red and blue. We're going to have yellow structures representing nervous structures, so sensory receptors and things like that. And we also have a lot of glands and hair follicles here. So overall, this layer is vascularized, so it's going to be providing nutrients that will diffuse into the epidermis or just hang out in this layer. It is also thermoregulating because of the presence of these blood vessels. So we can vasodilate or vasoconstrict flow to these parts of the body to help regulate that temperature. And we also have these sensory receptors. So this is where we feel things like light touch, deeper vibrations, of course, temperature and pain and all that good stuff. There are two main layers of the dermis. We have this papillary layer, which is more superficial, and then we're going to have this reticular layer. So again, don't be confused by the basement membrane. Don't let that trip you up. What we have here is this wavy pattern at the very bottom of the purple. That's going to be our basement membrane. We're going to have a thin layer of papillary dermis, which is mostly loose areolar connective tissue. So it's that less densely woven tissue that we talked about last lab. And then most of what we see here is reticular dermis. This is mostly dense, irregular connective tissue. So you can see we've got this swirly pattern here of those collagen fibers. The reason for this structure is that it provides strength and flexibility. So that's one of the reasons we can pluck at our skin, we can manipulate it, move it around, because we have this irregular pattern of connective tissue. If we take a look, so I'll be switching today back to the virtual microscope, we can see that same pattern here. So the stuff in purple is our epidermis, and then immediately deep to that we have a thin layer of loose areolar connective tissue, and then we have this really swirly change in texture. This is going to be our dense, irregular connective tissue. So it's a subtle difference, but it helps to recognize that our papillary dermis is really thin compared to the rest of the dermis. All right, we have glands as well. So this is where it gets tricky for students. Within the dermis and a little bit in the hypodermis, we have sweat glands. What we will be talking about today is exocrine glands. Exo means out. So these are glands that have ducts, D-U-C-T. Those are going to be secreting materials. So any sort of exocrine gland is usually associated with a duct. We'll be talking mostly about sweat glands and oily glands. So sudoriferous means sweat producing. Sebaceous is going to be more of our oily glands. We do have lactiferous or milk glands and earwax glands. Those are specialized. They're only found in specific regions, so we will not be dealing with them in this presentation. All right. So really the thing that is tricky about sweat glands is not identifying them, but just remembering the names. So for whatever reason, this naming system was always really confusing to me, so I'm going to try to make it clear for you guys. We have two names for most of these. So our most common type of gland is called a merocrine or an eccrine sweat gland. These are like your typical sweat glands you think about when you perspire, just exercise, you sweat. We're dealing with a merocrine sweat gland. We have apocrine sweat glands, which are a little stickier. They have a viscous secretion. These are usually responsible for body odor, and they're usually found in like smelly areas like the groin or the armpit, the axilla, and they're also associated with hair follicles. So that will be important. Remember that association. What's confusing is they both use merocrine secretion. This is a type of secretion, it's exocytosis, so things are being pushed out of cells through ducts to some sort of external landing place. In this case, with merocrine secretion, the gland is preserved. So there's some types where the gland is completely destroyed. We are just talking about merocrine secretion where the gland remains intact. So we have our two glands here. What this will look like on your model is a little different. So the thing to remember about apocrine sweat glands is they are associated with hair follicles. So on your model, if you're trying to identify a gland, figure out where its duct is going. In this case, we have a hair follicle. The duct is branching off and leading to an apocrine sweat gland. Compare that to a merocrine sweat gland on the right side. There is no associated hair follicle. It's just kind of free floating in the dermis. That's the main distinction when you're trying to identify these things on a model. And then we have sebaceous glands. These guys are a little different. So when sebaceous glands secrete stuff, the cell can actually be destroyed, or the gland, well, the cells of the gland can be destroyed. So think of like popping a pimple. Something is secreted, but at the end you don't have a full pimple. It gets destroyed in the process. These guys are associated with hair follicles as well, so they're responsible for making our hair oily. They look a little different on our model, so I'll point that out at the end. But what you want to look for is these clusters of cells that look like clusters of grapes. That's what I think about when I'm trying to identify them. And their name comes from the fact that they are secreting this really oily sebum. That's a type of material. It lubricates and it actually helps prevent bacteria. So let's take a look at that model. So we just talked about sebaceous. I'll start with that one. Sebaceous are going to have these clusters that look like grapes. So we're going to look for them near the epidermis. You can see they're pretty superficial. So on your model, find a hair follicle, and then we see this gland in gray with these clusters of cells. So this tells us this is a sebaceous gland. Our other gland associated with hair is going to be the apocrine gland. So again, let's find a hair follicle, follow it down, and then we have this guy. So we have a duct coming off that hair follicle, leading to a really large gland. This is an apocrine gland. So you can see it sits a little deeper compared to our sebaceous gland here. It has a duct leading to a hair follicle. And then by process of elimination, we have that last free-floating gland, the merocrine or the eccrine gland. that is not associated with hair. So that would look more like this guy here. Again, no hair follicle, or it looks like we have another one down here. So if you look at your gland and there is no duct leading to a hair follicle, or it is not sandwiched between a hair follicle, that means it is a merocrine gland. Okay. Pause a second. Do we have any questions about glands before we move on? Okay. So now we're going to talk briefly about nervous structures. I want you to be comfortable with this term corpuscle. That will pop up a lot. That's like a nerve ending, usually some sort of round nerve ending. Um, in this case, we have two types that we'll talk about. And of course, they both have two names because this is anatomy and everything has two names. So the first type is a tactile or Meissner's corpuscle. This guy is really superficial, so it's going to be found near the border between our epidermis and our dermis. So if we come back here, I didn't mention it yet, but we have a specific name for this wavy pattern made by the basement membrane. These little invaginations or dips, these are called dermal papillae. So papillae is like a finger-like projection. So within those papillae, we will occasionally have a tactile corpuscle, which is this little round yellow structure. So you can see they sit really close to our epidermis, so they are responsible for sensations such as light touch, really superficial sensory feedback. We have deeper corpuscles called Pacinian or lamellar corpuscles. I personally like the term lamellar, so a lamella is a layer. And to me, these corpuscles look like they have layers, like a fingerprint. So these guys are going to sit really deep in the dermis. They are responsible for deeper vibration sensation. So if we follow our model down towards the hypodermis, we've got these yellow guys here that look kind of like fingerprints. These are going to be those lamellar corpuscles. So you can use either name on the lab exam, full credit for both. Just make sure you're not getting the two confused. And here they are again. This image is available to you in your module, and I'll show you at the end the full key and what all you need to know for this perspective. But again, note the position of these corpuscles, whether they're superficial or much deeper, and that affects what sensations they can pick up on. Okay, hair. So sometimes I think our lab manual makes hair a little more complicated than it has to be. There's really not a lot of structures I can ask you about hair under a microscope because we can't get too much detail with our light microscopes. But overall, the hair is enclosed in a follicle. And that is this kind of pouch you see here wrapping around our hair cell. Most of the time on our slides, when we look at skin, if we have a hair cell, it can be fully sticking out. So that would be a type of hair cell we can visibly see on the surface of our skin. Sometimes we will get hair cells that are completely enclosed, so they're not poking out yet. But in any case, the hair will be surrounded by a follicle. This is interesting because the follicle is actually an extension of our epidermis. So while it looks like the follicle is going deep into our dermis, what we see is this little border of epithelium or epidermis in dark pink. So this is actually cellular. It's part of the epidermis. Overall, cell-wise, we have the same types of cells. So everything's keratinized. It has that tough protein providing a protective function. So our hair has three main parts. So we are no longer talking about the follicle. Now we're talking about the actual hair piece within the follicle. The part that sticks out is the shaft. So if you have visible hair on your skin, you're actually seeing the shaft of the hair. The root is going to be everything inside the follicle, so underneath the skin. And then we have this terminal portion, this bulb here, which is the alive part of the hair. So this is innervated and vascularized, which means if you rip out hair from the bulb, it really hurts, and that's why. So within the bulb, we have this structure, another little papilla, one of those projections. Within the papilla, we have these really tiny dermal blood vessels. So these are capillaries supplying the hair bulb. So I do want to note that the hair root is enclosed by the follicle. Don't get those two concepts confused on the lab exam. The hair root is part of the hair itself, whereas that follicle is that cellular extension wrapping around the entire hair. Usually associated with hair, we have a smooth muscle called the erector pili. And this is the tiniest muscle in the body. We have tons and tons of them, usually one associated with each hair root. And these are a little tricky to see on our slide, so we'll try to find a good example for you. But you can definitely see these on our model. So anytime you have a fear response, or if you get really cold, you get these goosebumps. Your hair stands on end. And so what's happening is these little muscles, these erector pili, are contracting to help make the hair more upright. And that is why we call it our hair standing on end. On a slide, they are a lot trickier to see. So basically, we are looking at this specific region. We've zoomed in really closely. We have a sebaceous gland. So those are the clusters of grape-like structures you see here. This is the lumen of a hair follicle. And then running next to it, we have this change in texture. So you can see these fibers are really smooth. They are still cellular because we have these dark purple nuclei, but what we're dealing with is smooth muscle. This is the erector pili. Now, I will not expect you to be able to identify the erector pili on a slide. So let's talk about what I could ask from a microscope. Obviously, we have the parts of the hair. So in this case, this hair is sticking out. So we do have a shaft, a root, and a bulb. This is another example of hair that is not sticking out. So I'm more likely to ask you from this typical shape we're expecting. Make sure you know the difference between the follicle and the hair itself. And then we can also talk about glands and stuff. So if you're looking under a microscope, usually we'll have some nice examples of sebaceous glands wrapping around the hair, in this case, the hair shaft or hair root. I could ask you about the dermis versus the hypodermis. So down here, we're getting into that layer of adipose tissue, which is why you see all these white puffy cells here. And then, of course, we have our layers of the epidermis. So a lot of stuff going on from a microscope perspective. Okay, let's take a look briefly at that virtual microscope, and then I'll show you the key for our skin model. So to review, we covered layers of the epidermis. We talked about the two layers of the dermis. So we've got that loose areolar connective tissue on top of dense irregular connective tissue. Make sure you know your Pacinian and tactile corpuscles and know the difference between the two and where to find them. Make sure you're comfortable with all of these structures and how to see them under slide. So there's some things that we talked about that are too specific under a microscope, but we can definitely identify our shaft, our root, our bulb, and follicle. And then make sure you hit glands hard because this tends to trip students up. Knowing where to find them on our model, whether they're associated with hair or not, and just some basic things about their structures. They're like whether they look like grapes or like a big cluster of tubes. Things like that are going to help you. So last two things for today. Let's zoom out again on our skin here. So again, up top we have our epidermis. That's going to be this darker purple cellular material. We have our swirly pattern of dermis with connective tissue. At the basement membrane here between the epidermis and the dermis is where we would find our dermal papillae, these little projections. And sometimes, if we're lucky, we can find those tactile corpuscles. I'm not sure if we'll find any on this slide, but you can use the PowerPoint. What I do want to point out here is hair. So this here is a hair follicle. You can see it is not completely sticking out from the surface. So in some cases, you will have hair like this that is premature, and it's not going to have a shaft yet. What we do have is, okay, this is calling it a shaft. So I would argue that because this hair is not sticking out from the surface yet, we're going to call it hair root and hair bowl. I would not call this the hair shaft on the lab exam because it is not sticking out. So just be aware of that technicality. Around the top of the hair, we have these clusters. So they should look pretty familiar by now. These grape-like clusters indicate that this is a sebaceous gland, and that does check out because it is associated with a hair follicle. And then another thing, if you zoom in really closely here, we can see this little itty bitty cluster of smooth muscle fibers forming an erector pili. But as you can see, this is really tiny. It's easy to get lost in the details, so I'm not going to ask you that under a microscope for the lab exam. So mainly hair, glands, and then things like the layers of the epidermis and dermis for the microscopes. And then last thing I want to show you is our model. So if you go to modules, go to lab 4. At the very top, we have this integument model, which is labeled. So if you click on that, it'll take you to this image. And this is everything I could possibly ask you on the model. So basically, you can make your answer key, write down all these terms, take a blank picture of the model, and try to identify all of them. See what you know. One thing I want to point out here is, again, when you're trying to identify your glands, think about whether they're associated with the hair follicle or not. Again, merocrine glands are free-floating. Think about where they are. So the sebaceous glands are more superficial. They're real close to that epidermis. Apocrine and merocrine glands are typically deeper. And don't forget about your sensory receptors, your tactile corpuscles versus your Pacinian or lamellar corpuscles. So use this key to help. But that is all I have planned for you today. So for the people that came in a little late, you do not need to complete the attendance sheet.